Very good morning, students. Today we are going to discuss the primary structure of monocot stem. Example, maize stem. So just see the picture. This is the morphology of a monocot stem. So we can see the sheath, node, leaf, inflorescence, internodes on the stem. So this is the external feature. Let me see the internal structure of monocot stem. So the outline of the maize stem or monocot stem in transverse section is more or less circular. Internal structure of monocotyledonous stem reveals epidermis, hypodermis, ground tissue and vascular bundle. So this is the ground plan of internal structure of monocot stem. Here we can see epidermis, outermost layer, then ground tissue and then vascular bundles. So monocot stem comprises three different types of tissue system: epidermal tissue system, ground tissue system, vascular tissue system. So epidermal tissue system, epidermis, cuticle, epidermal outgrowth, stomata. Ground tissue system, in the case of monocot stem, it is not differentiated into cortex, endodermis, pericycle, pith as in the case of dicot stem. It's a continuous mass of parenchyma cells. Vascular bundles with xylem and phloem, cambium absent in monocot stem. Let me see the internal structure of monocot stem in detail. Internal structure of monocotyledonous stem reveals epidermis, hypodermis, ground tissue and vascular bundles. First, epidermis. Epidermis is the outermost layer of the stem. It is made up of single layer of tightly packed parenchymatous cells. Their outer walls are covered with thick cuticle. The continuity of the layer may be broken here and there by the presence of a few stomata. There are no epidermal outgrowth. So epidermal tissue system constitute epidermis, cuticle and then stomata. So the major function of epidermis, protection, protects the inner lying tissues, cuticle, check transpiration, stomata, transpiration and gaseous exchange. So this is about the epidermis. Next to epidermis, we can see hypodermis. In dicot stem also, next to epidermis, we can see hypodermis. In dicot stem, hypodermis made up of collenchyma cells. But in monocot stem, a few layers of sclerenchymatous cells lying below the epidermis constitute the hypodermis. This layer gives mechanical strength to the plant. It is not continuous, it is interrupted here and there by chlorenchyma cells. So hypodermis of monocot stem constitute sclerenchyma and chlorenchyma cells. Next to hypodermis, we can see ground tissues. There is no differentiation into cortex, endodermis, pericycle and pith. The entire mass of parenchymatous cells lying inner to the hypodermis forms the ground tissue. The cell wall is made up of cellulose. The cells contain reservoir materials like starch. The cells of the ground tissue next to the hypodermis are smaller in size. polygonal in shape and compactly arranged, closely arranged without any intercellular space. Towards the center, the cells are loosely arranged, rounded in shape and bigger in size. The vascular bundles lie embedded in this tissue. The ground tissue stores food and performs gaseous exchange. So the major function of ground tissue, storage of food and gaseous exchange. Next, vascular bundles. So see the vascular bundles. Vascular bundles are scattered in the parenchymatous ground tissue. Each vascular bundle is surrounded by a sheath of sclerenchymatous fiber called bundle sheath. So what is bundle sheath? Vascular bundle is surrounded by a sheath of sclerenchymatous fiber. This is only called as bundle sheath. The vascular bundles are conjoint, collateral, endarc and closed. So what is conjoint vascular bundle? Xylem and phloem arranged in the same radius. Collateral, xylem and phloem arranged in the same radius, phloem facing outwards. Endarc, protoxylem points facing towards the center and metaxylem facing towards the periphery. This condition is known as endarc. Closed. So what do you mean by closed? Cambium absent in the vascular bundle. So this condition is called as closed vascular bundle. So in monocot stem, the vascular bundles are conjoint, collateral, endarc and closed. Vascular bundles are numerous, small and closely arranged in the peripheral portion. Towards the center, the bundles are comparatively large in size and loosely arranged. The vascular bundles are skull shaped. So this is about the nature of vascular bundle in monocot stem. In vascular bundles, we can see phloem and xylem. There is no cambium. So phloem. The phloem in the monocot stem consists of sieve tubes and companion cells. Phloem parenchyma and phloem fibers are absent. It can be distinguished into an outer crushed protophloem and an inner metaphlloem. Next to that, we can see xylem. Xylem vessels are arranged in the form of the letter Y. The two metaxylem vessels are located at the upper two arms and one or two protoxylem vessels at the base. In monocot stem, we can see protoxylem lacuna. So what do you mean by protoxylem lacuna? In a mature vascular bundle, the lowest protoxylem disintegrates and forms a cavity. This is only known as protoxylem lacuna. So this is about the xylem of monocot stem. So see the picture, transverse section of monocot stem, a ground plan and a sector enlarged. So it comprises three different types of tissue system: epidermal tissue system, vascular tissue system and ground tissue system. So epidermal tissue system, which include epidermis, cuticle, stomata. Then ground tissue system, not differentiated into cortex, endodermis, pericycle and pith. It's a continuous mass of parenchymatous cells. Their vascular bundles are scattered. Next, vascular tissue system. Just see the picture in the ground plan. Towards the periphery, the vascular bundles are numerous, small in size. Towards the center, the vascular bundles are less in number, loosely arranged, large in size. So vascular bundle with xylem and phloem. In monocot stem, the vascular bundles are skull shaped and the vascular bundles are conjoint, collateral, endarc closed. Then regarding xylem, protoxylem lacuna is present. This is one of the important characteristic feature of monocot stem. Phloem parenchyma and phloem fibers are absent in the case of monocot stem. So there is no pith. So this is about the transverse section of monocot stem. Now see the anatomical differences between dicot stem and monocot stem. A portion enlarged is given. First transverse section of dicot stem, next transverse section of monocot stem. Let me see what are the differences, anatomical differences between dicot stem and monocot stem. So in the first column, characters given. Hypodermis. So what about the hypodermis in monocot stem and dicot stem? Regarding hypodermis, in dicot stem, it is made up of collenchymatous cells. In monocot stem, hypodermis is made up of sclerenchymatous cells. Hypodermis provide mechanical strength. This is the first anatomical difference between dicot stem and monocot stem. Next character, ground tissue. So in dicot stem, ground tissue differentiated into cortex, endodermis, pericycle and pith. But in monocot stem, ground tissue not differentiated, but it is a continuous mass of parenchyma. Next difference, starch sheath. In dicot stem, starch sheath is present. In monocot stem, starch sheath is absent. Next, medullary rays. In dicot stem, medullary rays are present. In monocot stem, medullary rays are absent. Regarding vascular bundle, what are all the differences between dicot stem and monocot stem? In dicot stem, vascular bundles are collateral and open, but monocot stem collateral and closed. In dicot stem, vascular bundles are arranged in a ring, but monocot stem vascular bundles are scattered in ground tissue. In dicot stem vascular bundles, secondary growth occur due to the presence of cambium. But in monocot stem, secondary growth usually does not occur since there is no cambium. So these are all the anatomical differences between dicot stem and monocot stem. Just see the picture. Transverse section of monocot stem, ground plan and a sector enlarged. Then transverse section of dicot stem, ground plan and a sector enlarged. So what are all the important anatomical differences between dicot stem and monocot stem? Already we discussed. I hope you would have understand the internal structure of stem, dicot and monocot stem, and the anatomical differences between dicot stem and monocot stem. That's all about today's class. Thank you.